over to our moderator for today's session, Professor Hong Chen, to introduce the speaker. And just a reminder, please ask any questions for the speaker using the Q&A feature in Zoom. So thank you all again. Looking forward to the webinar.
Good morning. My name is Hong Chen. I'm an associate professor at Harvard Medical School, Boston Children's Hospital. It's really my great pleasure to introduce today's speaker, Dr. Liya Yin. Liya has an impressive training credential. So start with, she has an MD from Wuhan University from China. She also obtained a PhD from same university in China. Then she moved to University of California, San Francisco, UCSF, and did her postdoc. So since then, she transitioned to NEMED, Northeast Ohio Medical University, and she's there since. She's promoted associate professor since 2015. So other than this incredible training records, she also has a decorated accomplishment in scientific services. For instance, she's serving on the editorial board member of American Journal of Physiology, Heart and Circulation Physiology, and she's editor of future topic, Women in Cardiovascular Therapeutics, organized by Frontiers. She's also associate editor of Frontiers in Cardiovascular Medicine, and she's chair of fellowship committee and a member of steering committee of cardiovascular session in American Physiology Society, APS. And she's very active and also enthusiastic in the community service. She's been serving on the ad hoc members for a number of NIH study sections, and also active member of review member of AHA study sections. For instance, the Transformational Project Award, and also Career Development Award. So in addition to that, she's actually serving on the editorial board member of Circulation Research and the editorial board member of the International Journal of Cardiology. So, but last but not the least, she's on the editorial board member of this journal, Journal of Molecular and Cellular Cardiology. So then what's more, she has a number of awards and honors. And so most recently, she served as session co-chair, future topic, advanced inter-organ communications, just happening in experimental biology in Philadelphia in early this month. And she also a session co-chair of a symposium, signaling mechanism underlying myocardiovascular and microcardiovascular complications in type 2 diabetes, also happened in experimental biology. And she received outstanding faculty research award in 2020. And she also served the session chair in mechanotransduction, angiogenesis and remodeling in the 11th World Congress for Microcirculation back in Vancouver. So the list will go on and on. So for the time's sake, so stop here. But I like to introduce the scientific contribution from Dr. Lin's lab. Then with no doubt, we'll hear a lot more about her wonderful presentation and wonderful research topic just in a minute. But the research in Dr. Lin's lab centers on coronary collateral growth and microcirculation function and the regulation of coronary circulation and the myocardial blood flow in physiological and pathological conditions such as metabolic syndrome and heart failure. This this is her niche, also her pioneer work. And also she works on the stem cells in cardiovascular regeneration. And she used cutting edge tools such as multiphoton and light sheet scope, microscope, micro-CT for vascular imaging, and the contrast echocardiograph, and tissue Doppler for myocardial blood flow measurements. And I'm sure we'll learn and appreciate a lot more about beautiful images in this presentation followed. So without further ado, so with that, I'll hand this baton to Dr. Liya Yin. Thank you, Liya.
Thank you, Hong, for your nice introduction. So I'm going to share my screen and put about in the slide mode. Can you guys see well?
Yes.
Okay. Thanks everyone for joining this webinar. My name is Liya Yin. I'm an associate professor in the NeoMed. And I want to have the special thank to Dr. Tian and the editor board and give this paper accepted and being selected as the editor's choice. And I want to thank Dr. Van Gonnes for organizing this seminar, very serious. So basically we learn a lot through this online webinar. So today I'm talking about the ischemia heart disease. So the treatment of ischemia heart disease is still on unmet need because cardiovascular disease is #1 killer. And with the aging population and the increase of the prevalence of the risk factors such as obesity and diabetes, cardiovascular disease will continue to be a burden for the healthcare system. So most people, they can survive for the heart attack because of the bypass surgery, but most still have some suffer from the heart failure. So they're waiting just for the donation of the heart, but that will be very limited. So one thing, because I'm interested in the diabetes, so basically for the diabetes patients, if they have the ischemic heart disease, basically they have poor outcome than those without diabetes. So I know a lot of people work on the regeneration and ischemic heart disease. So we focus on the coronary circulation because we believe coronary circulation is critical for myocardial blood flow. So when exercise or stress, so the myocardial blood metabolic demand is increased. So you will need the more coronary blood flow increase to meet this like oxygen demanding. So if the imbalance of the myocardial oxygen demanding to the supply, then will cause the ischemia. So coronary arterial disease is well studied because a lot of people studying atherosclerosis because that occlusion in the big vessel then causes ischemia. However, recently more people are aware there's some coronary microvascular dysfunction. So that will cause the vasodilation problem. So they will have deficient of the ischemia perfusion. So that caused the heart failure and ischemia. So they have ANOCA. Basically have no obstructive coronary arterial disease or Takotsubo or the heart failure with preserved ejection fraction or diabetic cardiomyopathy. So my lab is focused on 2 goals. Basically, is one goal to prevent the stress-induced myocardial ischemia. So we have 2 sub-goals. Is like one, develop the coronary blood flow by stimulating the coronary collateral growth. So in case of you have the coronary arterial disease, you have the occlusion of the main branch of the coronary artery. So we can develop the coronary blood flow. So you can compensate the ischemia. The second one is preserve the coronary arterial dilation. So that will fix the coronary microvascular disease. So today I'm going to focus on the first one, how to develop the coronary blood flow by stimulating the coronary collateral growth. So, you know, when people have the heart attack, they have the ischemic heart. So the surgeon will open the chest, do the bypass surgery. So they will rescue the ischemia in the myocardium. So we believe the collaterals are the nature bypass because collaterals are the arterial connection. So when you have this arterial connection, basically can rescue the ischemia area by these branching vessels to perfuse the ischemia myocardium. So this is a human heart. Basically, you have the connection between the LAD and RCA. So this mechanism of collateral exists in the human, but not everyone have that. So for the people who have the well-developed collaterals, so they can survive better when they have the heart attack. So for clinically, people use the range of score to evaluate if the patient has have the collateral or no. So A is the 0. Basically, you have the occlusion of LAD, but there's no flow here, no collaterals here. So B is 1. Basically, you have the occlusion of the LAD, but there's small branch there. But the epicardial segment of the coronary artery are not shown up here. So if you look at the C, they score 2. Basically, you have the collaterals there, and there's some show up for the epicardial segment of the artery. So for these 4, basically, you can see 4 collateral the segment of the epicardial segment of the collaterals. So that's basically how people how the clinician evaluate the patient have the collateral flow there. So clinically, people also try to use the to promote the collateral growth for the ischemia disease. So basically, they can use the external coronal counter pulsation. Basically, they put a pressure cuff under the low limb, and so then they connect with the ECG. So monitor your ECG. So when in the cardiac systolic cycle, they deflate the cuff. So when you do the diastolic cycle, so they inflate the cuff. So then they have the shear stress to stimulate coronary endothelial cell to promote the collateral growth. So this is the outline of our talk today. So basically, we want to stimulate collateral growth. The first one we study is to use the stem cell therapy. So we basically reprogram the vascular and the aortic endothelial cells to the progenitor cells. Instead of fully reprogram the cells to the iPS cells, we do the partially reprogram the red endothelial cells to the vascular progenitor cells. So these vascular progenitor cells only can differentiate the endothelial cell in the smooth muscle cells. They cannot become cardiomyocytes. So instead of fully reprogram, most people we use inducible lentivirus system. So basically, we use the same for Yamanaka for transcription factor. So they are reprogrammed like iPS cells. But when we withdraw the doxycycline, so these ESL colony will become the progenitor cells. So after the in vitro assay, because it's already published, so we inject these cells in a rat heart. So basically, we have a occluder here. That's how we do the repetitive ischemia. When we inflate the balloon, 40 seconds every 20 minutes for 3 hours, and then that will rest 5 hours. So this cycle goes through until 10 days. So with this rat model of collaterals to stimulate collaterals, so we inject cells under this ischemia, we also call collateral dependent zone. Why is collateral dependent zone? Because when you inflate the balloon, they will cut the blood flow here. This red area will be ischemia. Unless you have the collaterals to compensate, then the cardiac function will be recovered or the myocardial perfusion can be recovered. So we inject the TD tomato labyrinth iPS cells and intramuscularly in the heart under this CZ zone. So you can see this red TD tomato cells, even though they are injected intramuscular, so they engraft well in the vessel. This green are the vascular wall, coronary vascular wall, and the red cells align there well. So how's the cardiac function? So basically, we measure the coronary blood flow from the NZ and the CZ. So without inflated balloon, there's no ischemia in this area. So the ratio from the CZ and NZ should be 1. However, if you inflate the balloon, so if there's no collaterals, there will be 0. So in this rat repetitive ischemia model, when we the rat underwent the repetitive ischemia, you can see the ratio from CC to NC is 0.5. That means the collaterals developed by this procedure can compensate 50% of the ischemia induced by inflation of the balloon. So compared to the endothelial cell, mesenchymal stem cell, and fully reprogramed iPS cells, these vascular progenitor cells, they can increase the blood the ratio from 0.5 to 0.8. That means this stem cell can promote collateral growth. So basically, even you cut the blood flow here, there are 80% of the myocardium is being perfused by this collateral flow. So to make our study more clinical translation, we apply the cells to the Zucker obesity rat. That's a rat model of the metabolic syndrome. So as we see here, for the lean animal, the progenitor cells work very well, but look at the Zucker obesity rat. So basically, without intervention, so they have less collaterals than the lean animal. So even you inject the progenitor cells, they didn't achieve as much collaterals as the lean rat. So we feel, okay, maybe endothelial cell is not good enough. Maybe smooth muscle is more potent because endothelial cell is more vulnerable for the damage. So we use the same approach, reprogram the smooth muscle cells. So these reprogrammed small muscle cells have very potent angiogenesis potential. So this is a 2D culture. So this reprogrammed the smooth muscle cell can become endothelial cell, and the smooth muscle cell is a 2D culture. They have the branching and the network, suggesting these reprogrammed smooth muscle cells have very potent angiogenesis potential. So we inject these cells in the obesity rat. So basically, they are performing better than the reprogrammed endothelial cells. So then when we said, okay, we try to treat them, but then we step back to see, we really don't know what's the mechanism for this collateral growth, because large animal didn't give us enough tools for genetic modification, linear tracing. So we feel, just like compare the Zucker lean and Zucker obesity one, and the gene expression profile. So basically, surprisingly, compared to Zucker obesity and the Zucker lean during the collateral growth, the most cluster signal cluster is like a regulation of the apoptosis and cell death. So we also do the RNA-seq, compare the Zucker obesity one, Zucker lean one, that's the genetic model obesity rat. So we also use a high fat, high sugar for 6 months to compare the lean animal with the diet-induced model. Surprisingly, microRNA 21, they are like overexpressed both in the obesity one, in the Zucker obesity one and the diet-induced obesity one. So we validate the gene expression in the collateral dependent zone. So Zucker lean and Zucker obesity MIR21 have high expression, and this high fat, high sugar diet treat obesity rats have the high expression MIR21. So MIR21 has been studied for a while, and they have the different roles in the cardiovascular disease. But I have to say they're very cell and specific and disease specific. So to address the MIR21 in the collateral growth, so we need a mouse model for collateral growth. So basically, we have to scale down from the rat to the mouse. But as you know, mouse heart is 1/10 of the rat. So it's a little bit harder to do the balloon surgery, and the tether system is like have to be adjusted. So eventually we successfully have this mouse model. Basically, we do the put a balloon there. So then this balloon will be inflate, deflate. Different from the rat, we have to do the 17 days to develop the collaterals. So this is a microfill heart. And the yellow is the microfill polymerized after 30 minutes. So this is a balloon over the LAD. The blue stitcher is like how you tie the balloon here. So with the inflate balloon, deflate balloon, we have the repetitive ischemia in the mouse. So this is the rat heart, the mouse heart without RI. And this is the right mouse heart with RI. You can see even the cut of the ligated LAD, these below vessels are being filled, suggests the collaterals grow under this area. So this is just a micro CT imaging, and you can see there's some connection between the RCA and to the LAD. So if we look like 3D micro CT imaging, this is a naive heart. You can see the LAD, RCA, and the septum. So then if you ligate LAD, that's the same situation when people have heart attack. Same for the MRI model, have ligating LAD. So you have zero perfusion here. So that suggests the mouse does not have the native collaterals. So this the imaging shows like the mouse heart with the RI. So you ligate here, so you still see the perfusion here through this like a purple and yellow collaterals. So we've also do the tree analysis to see these branches of the vessel. So basically this is the white type without RI. This is the white type with RI. You can see they have more branches and end branches between the white type with the RI and without RI. If you look more carefully from the like the map of the left arterial and the septum and the right, you can see after repetitive ischemia, you have more collaterals. So they are branching more. So then we want to say, okay, we have a good model for collaterals. We can really address how the collateral grows. So as we said, the big traditional dogma is like the collaterals are enlargement of arterial connection in the heart. So there are some new study from Dr. Jim Faber's group shows like then when you do the distal ligation of the LAD, they see some collaterals there, this de novo collateral formation. Chris Redhouse and the lab, they also published a paper. So basically their theory is like an arterial endothelial cell reassembling for the neonate collateral formation. And for the adult, you have used the CXCL12 to stimulate the collaterals. So we also found like Dr. Binzo has a paper and have the very unique tool for the Appling linear tracing. In their study, the Appling is labeling the sprouting capillary endothelial cells, and they didn't find Appling positive endothelial cells become arterioles in the MI model. So we go ahead because we think MI model and our repetitive ischemia model are very different, because MI model had an injury heart, they have tissue lost, they have all the injury signal. So we go ahead and use this Appling query to use for our collateral development study. So basically we have the surgery, put a tamoxifen injection, and let the mouse recover for 1 week, then we start the RI. Different than the Dr. Binzo study, because they only do 5 days in induction. So because Appling is sprouting, that if that occurs continuously, we just add more lower dosage tamoxifen or the tamoxifen food to maintain, we make sure catch all the sprouting endothelial cells. So that's our difference from the previous study. So we also look at the pan endothelial cell type 2. So that's the 1 millimeter heart slice, the confocal, the multiphoton image. So basically, pan type 2, we're labeling all the cells, big, small, capillary, or the arterial vein. So, so basically, we don't know which is new, which is old. So then we feel, okay, this is not what we need. So we go ahead and use the Appling query. So this is a slice of the heart. You can see sprouting endothelial cell there. And if you zoom in, the green cells specifically labeling the sprouting capillary endothelial cells. So we do the early stage of the imaging. So basically, this is the normal zone. As we said, that's above the balloon, and wouldn't have an ischemia during the inflation of balloon. So we have a collateral dependent zone. So when you inflate the balloon, there will be ischemia. So that's the NZ and the CZ. So we can see with the NC, you have a little sprouting endothelial cells, but for the CZ, you have more. So we want to ask if this sprouting endothelial cell proliferate. So we inject EDU to labyrinth proliferate cells. So basically, you can see the we use the anti-GLP to show the airplane positive cells, and the EDU is proliferating cells. Basically, you can see the co-localization of the sprouting and airplane cells and the EDU staining. If you quantitate the control in the RI, basically you have more sprouting, more proliferation in the CZ area in the repetitive ischemia model, and the EDU and the airplane double positive cells are more in the our RI model. So then we want to see if the later stage of the if this endothelial cell lasts long for the whole collateral growth. So basically we do the double layering. So we have the same like EDU 3 to 5 days, then have the 8 to 10 days to the BRDU layering. So you can see basically you have much less EDU layering cells during the late stage. So basically we feel like a sprouting capillary endothelial cells sprouting first, but later on they kind of like wind down. So then we do the imaging for this at the later stage. So basically these sprouting endothelial cells can expansion. You can see in the RI group and the vessel, big sprouting capillary, the expansion in the diameter and in the length. So this like we also want to make sure this like this airplane cells is expression as like before. So basically they are colocalization. These airplane cells are endothelial cells. So then this is just an imaging of the network of the airplane positive capillary cells. So basically we measure the diameter. We know like a micro capillary is like below 10 micron, right? So these new vessels, they are like the diameter is 20 to 30. So we just as a reference for the naive mouse heart is the septum arterial. So the branch is like 20 and 30. So basically this capillary can expansion to the arterial size. So we also, when we look at the images, you can see here, so we can see the green cells always align with the red cells. We know green are the airplane positive cells, red can be the other cell type. So basically we use the smooth muscle after acting staining the smooth muscle cell. We see this like small cell align well outside of the airplane positive cell. That means these smooth muscle cells participate on the collateral growth. So basically also suggests this capillary arterialization with the smooth muscle coverage. So we also do the want to see if that hypoxia contribute to these collaterals. So we use the hypoxia probe to labyrinth the hypoxia cells. Basically, we compare to the control group, the repetitive ischemia treatments cause more hypoxia in the CZ zone. So we also doing the gene expression in the mouse heart with the control and the sham and RI group. So you can see CXR4 is upregulated, and VEGF2 is upregulated, and JAK1 and HIF1 alpha is upregulated. So compared to the RI and the control, MCP1 is upregulated, and compare the control CZ zone and the RI CZ zone, HIF1 alpha is increased. So that means this fits in the literature. These factors contribute to the collateral growth. So then we have the question about cardiac function. So as I said, when you inflate the balloon, cause ischemia, if you have more collaterals, that means your heart will not so ischemia, so that won't change the cardiac function. So here just like shows the ejection fraction and the fraction shortening before the RI and after RI. Before RI, there's no collaterals here. So after RI, we're supposed to have some collaterals. So when you inflate the balloon at the beginning without the collaterals, so you will have the ischemia, cardiac function will decrease. So we compared before and after percentage change with treatment or without treatment. Basically, if you without before RI, so without collaterals, you have cardiac function in decrease 20%. But when you have collaterals, so you have ischemia because this collateral compensate the ischemia. So the ejection fraction is dropped to only like 5 or 10%. So basically, these collateral are functional because this provide collateral flow during the ischemia. So we also measure the coronal blood flow. So basically, that's just we inject the micro bubbles through the pelvis. Then we'll measure these micro bubbles during the destruction to see how this area have the micro bubbles. So if you have no blood flow here, so we will have less intensity. So basically, we measure the same of the CZ and NZ which we have here, but you just use it in different tools. So basically, we can see this vascular wall, the myocardial wall has been filled with the microbubbles. We can measure the blood flow. So if we compare before and after RI for the CZ to NZ as shown here, before the RI, the CZ to NZ have 20% compensation for the ischemia. But when you have the end, so basically you have 50% compensation of the blood flow. So this just use the contrast echo to measure the coronary blood flow. So we also just have the light sheet scope to show this is the type 2 heart with the RI for 17 days. So basically you can see all the vessels there, so big or small. We also have the airplane Cree. So basically these were more specifically labeling the new vessels there for the collaterals. So here's a summary for this part. Basically, we developed the mouse model for collateral growth, and we found these capillary endothelial coronary capillary endothelial cells can sprouting and expansion, and then can be mature and arterialization to become coronary arterioles. So as I mentioned, I want to interest in the metabolic syndrome for the collaterals. So basically, this is a diabetic heart. So basically, you can see the hypertrophy. That's the normal, not invention heart. So this is the acute. So we ligate on the AOAD. Then you have see no blood flow here. So it's this consistent with the literature for the diabetes rat mouse. We ligate AOAD, and you didn't see collaterals there, even though they have the RI stimulation. Basically, they confirm the impaired collateral growth in the diabetes. So then, as we said, okay, we want to restore the collateral growth by the gene. That's the whole goal for the mouse model, right? So then we go back to look at the miR-21. Basically, we see the miR-21 is upregulated in the white type and the DB, heart and aorta. So, and the coronary endothelial cell DB is miR-21 also is increased expression in the diabetic coronary endothelial cells. And when we use the human coronary endothelial cells, you treat with the high glucose, miR-21 is upregulated too. So this is the NS scope to see the miR-21 expression in the diabetic heart. So miR-21 is upregulated in endothelial cell too. So we go ahead use the miR-21 knockout mice. So this like this staining shows miR-21 expressed in the vascular aorta in the more small branch. So we do the white type miR-21 global knockout. So with ligation, they didn't have any collateral growth. So, but then we do the RNA with the miR-21 white type. They have the collaterals shown in purple here. So if we put the miR-21 white type on high fat sugar, again, like the diabetes animal, they don't have the collaterals. So, however, if we put miR-21 knockout on high fat sugar, you can see the bottom is being filled, suggests collaterals there. So this is microfield imaging. So basically, this is the white type with a high fat, high sugar, knockout with the high fat, high sugar. So miR-21 knockout rescued the phenotype of the impaired clot growth in the adult-induced diabetes. So here's a summary of my talk. So basically, we try to use stem cell to promote coronary clot growth, and we also try to understand what's the mechanism for clot growth. So we use linear tracing to see the process of the clot growth in the mouse model. So, and we found the miR-21 is upregulated in the diabetes and the metabolic syndrome. So we know how miR-21 can restore the collateral growth in the adult-induced diabetes mice. So the ongoing project, so basically we're going to do the single cell RNA sequence for these apelin positive cells to see what they are and at a different time point. And we also want to do the smooth muscle and immune cells to induce the to see if we induce both muscle linear tracing to see if smooth muscle contributed to clot growth. So we use some smooth muscle 22, and myeloid lineage lysozyme, we found it's very leaking. So basically that's not ideal for the two study. We are going to have the new mouse line to study that one. And the next goal is like to study, because we see the miR-21 global knockout rescue the phenotype, but we are going to use the endothelial knockout because they start from the endothelial cell sprouting, right? And the immune cells in the collateral growth. With that, I want to thank my lab people. So Anurag and Cody performed the experiment or the study, and Wei Guo is the postdoc who made this mouse model, so make all the study possible. So Sophia, Devin, and they all work on the micro-CT, and we have James, the surgery, and Molly is our lab manager and do all the mouse breeding. We have Yang and Alisa work with us, all make contribution to this one. I also want to thank Dr. Chilian, who is my department chair and who guided me through this collateral circulation. And my colleagues, Dr. Wahanian, who is the person who has this talent surgeon and do all the contrast echo and train my people. And Dr. Feng Dong is the other colleague. So I want to thank Bing Zhou, who is very generous, gave me the mice and participate in all the discussion. I just text him some of the questions. Of course, I want to thank my NIH funding for the project. Thank you again, and I'm happy to take any questions.
Great. Yeah, it's a wonderful presentation, beautiful images, and pretty sure everyone is impressed by how beautiful how powerful this light sheet microscopy can provide. So if you have any questions, please type in the chat or Q&A so I can pass to Liya. And I can start actually yes. So let me start with entertain the audience first. So Hua Zhu said a very elegant study, and congratulations, Liya. So his question is whether you have compared your RI protocol with the traditional ischemia preconditional protocol for coronary collateral growth.
Hua, that's a good question. We didn't do that because the precondition basically can can help protect the ischemia, but we didn't do that. Yeah, thanks.
Okay, so so while waiting for other questions, I throw a lot of congrats, wonderful talk, many congrats from Wan Xin. So what is the source for this collateral growth, the EC? Where's the EC coming from? I know it's a hard, it's still in a hard debate topic. Is it from the endocardium, or it's just the angiogenesis from pre-existing collateral vessels?
So based on from our study, because that's the other limitation for this study, because the mouse model, they don't have native collaterals. So like a pig doesn't have, the rat has, and the guinea pig have a lot. So so we for the dog, they have the pre-existing like the human. So in that case, in our study, we didn't have the pre-existing artery to expansion, but we start from scratch, basically it's from the capillary. Based on our study is from the capillary, not from endocardium. So I think Bing has the recently published for the endocardium, the two SV population. So for our study, we don't know because we just tracking the capillary endothelial cells. What's the source? Maybe there's some bone marrow or some resident stem cell participate this like capillary sprouting. We don't know.
Okay, great. Yeah, thank you. The other question from our audience is from Ma Yonggang. Great talk. How do you know if the newly formed vessels are really functional?
So that's a good question. So because when most people do the angiogenesis by the isolated lectin staining, PCAM staining. So first evidence is the micro CT. So we use microfield to perfuse this heart. So if there's no function blood flow, the vessel is not function, the microfield won't go through. So if the microfield being filled means this vessel are functional. That's the first evidence. Second one, we do the echocardiograph, right? So when you inflate the balloon, if there are no collaterals, then heart will be ischemia. That will be cardiac function will drop a lot, like the MI. So during our inflation after the simulation, the cardiac function didn't decrease as much as the MI. That means they have collaterals there.
Great. So thanks, Liya. Question from Eugene. Very nice study, Dr. Yin. One naive question: does LAD ligation induce collaterals? If not, have you compared RI versus acute ligation in terms of metabolic inflammatory MCP1 and HIF1 alpha?
Hey, Jun, thank you for good question. So basically, as I said, from Chris Redhouse study, so for the adult, they do the MI model, they didn't have the collaterals unless they intramuscularly inject the CXR SDF1. So for Jim Faber's paper, so they cannot do the big ligation. They have to do the distal, very bottom near the apex, have the small ischemia or the infection, then stimulate collaterals. So in our study, we actually did see, it's not all time, for the 3 months, we see the collaterals in the MI model. But again, the trick is like how big is the infection size will be. If too big, you have the injury, they won't have the collaterals. When you have the small, like a Jim Faber's paper, then maybe have some collaterals. So I try to compare this one, but it's really tricky when you have the big size of the infection or the small size, we don't know. And to address your question about MCP1 and HIF1 alpha, so Jim Faber published the other paper. They just put the mice under hypoxia. They can just develop collaterals. But however, that collaterals won't last longer. When you move back to the normaxial condition, this vessel will regress. So hypoxia is important for these collaterals. And the inflammatory MCP1, yeah, from Jim Faber's paper said it's important for the collateral growth. So again, they are doing the MI model because in MI, you have more inflammatory for our study because we have the surgery, we rest for 1 week, then until the mouse cardiac function go to normal, then we started on RI. So for our study, maybe we'll have less inflammatory, but our MCP1 is increased compared to control and the RI. HIF1 alpha should be the signal there. Yeah, thank you.
Thank you, Liya. Then, so a question from Dr. Jiang Xi. Excellent talk, congratulations. My question is whether the miR-21 dependent inhibition is NO or CO dependent.
That's a good question. So we published the other paper in the break. So basically, we studied the miR-21 knockout on high fat, high sugar for the vasodilation. So we know the white type or the normal vessel, the vessel dilation is NO dependent. So when in the diabetes, we found the vasodilation mediator is become a switch from the NO to hydrogen peroxide. So however, MIO-21 knockout, they can rescue that phenotype. The vasodilation go back to be NO dependent. So we didn't test the CO dependent yet. Maybe that's interesting to look at because we have some conditional knockout. The story didn't fit well. So maybe we should look at the CO. Thank you.
Okay, yeah, thank you, Liya. So Kai-Fu Chen has a question. Great talk, Liya. Are you able to distinguish between cells derived from the injected cells and the other cells of the same cell type in single cell RNA-seq data? And then are the injected cells, that means that the stem cells, your IVPS, from the same genetic background or different genetic background? Does this matter regarding the effect of the injection of the IPS?
Kai-Fu, thank you for a good question. So yes, we dropped that stem cells research because of the episode of that cardiac stem cell, whatever. So, but I still have the interest for that one. So we, because that reprogrammed cell is a heterogeneous. So we should be able to do the single cell sequence to see which contribute to collateral growth, or more angiogenic, right? So and we gave the cells to the other investigator. They found the exome from this IVPC, the progenitor cells, have the perichromic effect to promote the collaterals in the high limb. So basically, yeah, we didn't do that. It's still keep in my mind, then we can compare the single cell of these heterogeneous cells. And to answer your question, why we inject these cells in the zygobesity one. So basically, when the cells have been reprogrammed, so basically they lost the immunity for the other, the original one. So basically, we reprogram is the SD. So why we inject the zygobesity one? So we do the cell injection to see the spleen immune cells. There's no rejection for different strain, the stem cell. Basically, reprogramming erased the immunoreaction for the reprogrammed cells. Did that address your question? Yeah.
Okay, so thank you so much. The question from Halokashi Yadav, nice talk. Is any role of vitamin D in the collateral growth?
That's a good question. We didn't study that. Yeah. Sorry, yeah, we didn't study that.
Maybe you can try, just with some vitamin D. Okay, thanks. Another question from Zhou Guilin. That a great job, Liya. Congratulations. How small arteries, how small arteries do microfilament can go?
So microfill can go anywhere. But if we don't, what we want to see collaterals. So basically, you don't want to, when you do the perfusion, you have control the time and the pressure. Don't overfill them. Otherwise, all the vein or the capillary will be filled. So you won't be able to see the arterial.
Okay, so great. So thanks. So question from Q&A from Bruno Mocate. Nice talk. Is there a specific age where you have an increased probability for collaterals after ischemia, or the same level of collaterals happens both in pediatric and adult models?
So that's a good question we discussed in the paper. So basically, we didn't do the age comparison because this surgery is very challenging. So we try to control the time window. And we did, answer is no, we didn't do the age dependent. But I believe the all the cell regeneration and the stem cell will rely on the age. It is age dependent. So we didn't look at that yet. Maybe to a certain point when we can improve our surgical procedure technique, we can do the old one, because old mice, we don't know if they can survive that one. We did some old mice and we don't have good luck. So
yeah, thank you, Liya. a question from Sophie Miller. Thank you for a nice talk. I was wondering if you have looked at stress, like exercise, and how it affects the collateral sprouting, if it affects it the same way or the same amount of sprouting.
So exercise, people have the model for exercise to increase the coronary flow reserve, improve the cardiovascular function. We didn't, because because you have the tether system tie on your on the mouse neck, and then you have to go to the treadmill. So we didn't do that model. So, but I will expect if you have exercise, we improve the coronary blood flow. Maybe there is some improvement for the collateral growth. Thank you.
Okay, so thank you. Question from Dr. Yuhuan. Thank you, Liya. Is it possible to visualize the number of layers of vascular smooth muscle cells surrounding the newly formed coronary collateral following repetitive ischemia?
Dr. Huang, thank you. This is a good question because, as I said, we do the muscle staining is there, but we didn't count them how many layers. So that's why we use the muscle 22, but it's very leaky because I have cardiomyocyte something. So we hopefully will have new tools inducible smooth muscle will can address that question. Then we have to zoom in to see how many layers there.
Okay, great, thanks, Liya. So question from Rosie Wu. Congratulations, Liya. Impressive data and a beautiful presentation. Does the miR-21 level change in response to IR or any other diseases?
Rosie, thank you. This is a good question. We didn't check. Yeah, we didn't check. Maybe it's worthwhile to check. Thank you.
That's great. Thanks, Liya. A question from Renbin. Excellent research. Have you looked at the proliferation and differentiation of vascular endothelial cells in your model system? Proliferation you did, right? So.
Yeah, we checked the we checked the airplane positive cells. We can go back look the other cell type. The problem is that MTMG you only have 2 colors. So then we have the third color is purple. The answer is like we didn't check carefully, but we have the data. We can go back because we use the 647 selecting to staining. We can see the red and the purple, and that will address all the EDU, BRDU to see the vascular endothelial cells. You meant the arterial endothelial cells, right? So yeah, we can go back to look. Thank you.
Okay, great. Thanks. Yeah. The question from Zhao Guilin. Does apelin exist in other cell types besides new endothelial cells?
Based on thanks, Guilin. Based on Bing's data, the linear tracing is like a capillary endothelial cells. But I believe people use like apelin for the cancer sprouting endothelial cell too. So when they do the tumor, apelin is expressed in the tumor endothelial cells.
Okay, so that's excellent timing. So excellent question and the engagement of the audience. I'd like to really take this chance. Wait a second, there's a long short one. Okay, so there's a question, not the last, so we'll see. From Xiaochuan, excellent study, exciting study, congratulations. Beyond the miR-21, is miR-21 function exclusively EC dependent?
So Xiaochun, thank you for the question. So we have the endothelial cell knockout. So basically endothelial cell knockout basically in our primary data is phenocopied global knockout. We also have the other line like a myeloid lineage, and we also have smooth muscle lineage. So we didn't see much in the smooth muscle lineage. For the myeloid lineage knockout, it's like a work in progress. So we have to do more experiment to draw the final conclusion. Thank you.
Great, yeah. Okay, I'd like to really thank all the audience, and first thank the speaker for her wonderful and beautiful presentation. We really enjoyed a lot, and thank the audience for your great enthusiasm and participation. and excellent questions. So like to conclude with this session and say goodbye to everyone. Have a nice weekend. Bye-bye.
Thank you, Hong. Thank you, everyone, for your good question, for your time to attend this webinar. So thank you so much. Thanks, Ron.
Bye-bye. Yeah.